Hello, everybody. My name is Virginia Kaklamani from UT Health San Antonio. Welcome to this year in review. This has been a very popular session, so I appreciate everybody being here today. Our first talk is going to be our basic science year in review, given by Dr. Heide Ford from University of Colorado. Heide.
Okay. Hello, everyone. As was just mentioned, I'm Heide Ford from the University of Colorado, and I wanted to thank the organizers for this chance today to give you a review of the basic science that's occurred in the breast cancer field in this past year. I have no financial relationships to disclose. Okay, so I will say that when I was asked to do this, I didn't realize I don't know why I didn't realize, but I didn't think about what a daunting task this is. If you look in the literature, and I'm sure the number's higher now because I looked at a few weeks ago, there were as many as more than 26,000 actually breast cancer papers in PubMed just in this year. And so it's very difficult to decide what are we going to focus on when I wanted to give a review of the basic science that's occurred this year. And so what I ended up having to do was, first I'm going to apologize for anybody whose amazing science I'm not talking about today. There's a lot that I cannot cover in the 30 minutes I have. But I decided to break it into a few different areas. The first thing I'm going to talk about is some new insights into the biology of breast cancer subtypes. I'm not going to cover all subtypes either. The second thing I'm going to talk about is the importance of the innate immune microenvironment and the real discoveries this year that lead to us thinking about how to combine innate and adaptive immune therapies. And then I'm going to talk about a topic that I think comes up every year in the reviews of basic science, and that is why is it so difficult to treat metastases? This is still an incredibly difficult problem, and I'm really going to focus on tumor cell plasticity, focusing around epigenetics, but then an area that I think has become very hot in the last year and is really something that's been ignored for a lot of years, and that is mRNA and unique mechanisms of mRNA translational control. And in the last few minutes, I'm going to talk about some resources that would be valuable to everybody in the breast cancer community, basic scientists all the way to clinical researchers. Okay, so let's start with our new insights into the biology of breast cancer. So the first thing I'm going to study or talk about is a study in which the main question they were asking was not about breast cancer subtypes, but was instead about what the origin is of oncogenic copy number amplifications in breast cancer. And so in this study led by Peter Park's group, they wanted to understand really how focal amplifications, which we know are very common in breast cancer and turn on genes or upregulate genes like cyclin D1 and ERBB2, et cetera, actually occur. And so they used whole genome sequencing, RNA-seq, epigenomic data, and put it all together in order to identify a very novel mechanism that underlies focal amplification, and that mechanism they termed translocation bridge amplification. And that's a pretty complex diagram up there, and I can't point to all the steps from here, but what I will say is that it differs from what we typically think about, that's breakage fusion bridge cycle for the translocations and amplifications, and is instead actually caused by the amplification arises by these interchromosomal translocations that lead to dicentric chromosomes, a bridge formation, and then the breakage. The way this actually falls, though, into ER positive or hormone receptor positive breast cancer is because we've known for many years that estrogen induces DNA breaks. And what they showed is that the estrogen-induced breaks often occur near the target genes that are amplified, and that is followed by a repair that requires this interchromosomal translocation and ultimately results in the focal amplifications. So the interesting thing about this is the team then looked pan-cancer. So does this happen in a lot of different tumor types or different subtypes of breast cancer? And what they found is that this mechanism has real tissue specificity. So this type of interchromosomal translocation leading to amplification only occurs in a subset of cancers. And of note, while many years ago, actually the androgen receptor was shown to also be important for amplification, the mechanism utilized in hormone receptor positive cancers where the hormone is estrogen and ER is actually different than the mechanism used in AR-related cancers. So why does this matter? I think this matters just for how we think about what estrogens and estrogen receptor signaling does in ER-positive tumors. So we've known for many, many years that lifelong exposure to estrogens is a risk factor for breast cancer, and that, of course, we know that one of the reasons that therapies work that target this pathway is because estrogen and estrogen signaling is important for proliferation and for survival, preventing cell death. But this study lets us appreciate that there are additional other major mechanisms that contribute to how estrogen and estrogen signaling through the estrogen receptor contribute to cancer, and that is through altering the genome structure and leading to this translocation bridge amplification. The second study I'm going to talk about is also in the context of hormone receptor positive cancer. So this is in hormone receptor positive HER2 negative cancer, which we've heard a lot about at this meeting already. And what this group, led by Dr. Shao, was interested in asking was, can we improve targeted therapy specific to this subtype of breast cancer? And the reason they were so interested in that is, of course, as we all know, that 2/3 of breast cancers are hormone receptor positive and HER2 negative. But even though the prognosis might be better in this subset of cancers, there's still a persistent risk of relapse, a high intratumoral heterogeneity, and a resistance to hormone therapy. So this group performed DNA and RNA-seq metabolomics and proteomics. So they did a very widespread sort of omic analysis of hormone receptor positive, HER2 negative breast cancer. And they identified 4 additional subtypes within this class of breast cancers that contain the canonical luminal, a proliferative subtype, an immunogenic subtype, and an RTK-driven subtype. They then went on to develop convolutional neural network models using deep learning in order to infer the subtypes from digital pathology, with the idea that ultimately we might be able to use the digital pathology as a way to determine treatment options in these patients. Because what they showed is that these different groups can actually help inform different treatments we might give. So we know that a subset of these breast cancers are susceptible to CDK4 and 6 inhibitors, but they also show that there is a subset that's immunogenic. They have larger immune filtration than other of the hormone receptor positive HER2 negative breast cancers. And finally, they showed that there's a class that interacts quite substantially with the stroma and with the cancer-associated fibroblasts. Those fibroblasts have activated RTK signaling, and if you inhibit that signaling in that class, you actually inhibit the tumor growth. So again, this study was important for coming up with novel ways that we might be able to treat subsets of breast cancers within the hormone receptor positive HER2 negative class. Okay, we're now going to move on to triple negative breast cancer. And in this study, the main question was, why are immune checkpoint inhibitors ineffective for so many triple-negative breast cancer patients, and how do we overcome that? And this study, led by Justin Bulko, focused on triple-negative breast cancer, of course, because it has a high mutation rate and is expected to be highly immunogenic, and that although PD-1 is an approved therapy for the PD-L1 positive metastatic triple-negative breast cancer in combination with chemo, we still see that only about 15% of patients respond to this treatment. And so what this group did was single-cell RNA-seq and spatial technologies to look at intertumor heterogeneity in MHC1 in multiple types of breast cancer, actually, not just triple-negative breast cancer. But what they found is that MHC1 was very heterogeneous, particularly in the triple-negative breast cancer subtype. And of course, we know that MHC1 is important for the response of CD8-positive T cells to the tumor. So what they also observed in this study was that the heterogeneity of MHC1, so if it was not expressed on all the cells but in subsets of cells within the tumor, actually correlated with a high percentage of NK cells in the tumor. And so they developed these really beautiful mouse models where they enforced MHC1 expression, but in a heterogeneous manner, so not in all the cells within the tumor, and found that similar to what they had seen in the human patient samples, they found an increased influx of NK cells in those tumors that had heterogeneous MHC1 expression. And so this led them to the idea that they could try to activate the NK cells in these tumors that were non-responsive to PD-L1 therapies and then sensitize them to PD-L1 therapies. So they did exactly that using an antibody against NKG2A to activate the NK cells, and found that that could overcome PD-L1 resistance and increase survival. So this is a really novel and unique study that might provide a new way for us to think about how to overcome resistance to PD-1 therapies, in part by looking at heterogeneity and then activating the innate immune response. Okay, so that segues very well into the next section of this talk that I want to give, which is around the innate immune microenvironment and some really interesting findings in that space in breast cancer this past year. The first paper I want to talk about was a paper in which we were, the group led by Dr. Vignali was trying to look at the immune landscape, specifically of hormone receptor positive cancers. And the main question was just, can we learn more about the tumor immune microenvironment in these ER positive breast cancers? And can we differentiate between invasive ductal and lobular breast cancers in order to identify clearly actionable new targets? And so the reason this is important is as many of you know, historically ER-positive tumors are thought to be immunologically cold, and that's due to a lack of influx of T cells compared to triple-negative breast cancer, for example. And so what this group did was take samples from both IDC and ILC and then did both phenotypic, well, a number of analyses, phenotypic, transcriptional, and functional analyses for treatment-naive IDC and ILC. What they found, actually, that was interesting is that macrophages rather than T cells are the predominant immune cells in ER-positive breast cancer. And interestingly, these are also the cell type that are the most transcriptionally diverse between IDC and ILC, showing that there are differences in the immune response to these two diseases. They further showed by their spatial profiling that an interplay between macrophages and T cells is associated with longer disease-free survival, specifically in IDC, but not in ILC, again, pointing out that we need to treat these two diseases differently. Most importantly, the study is highlighting macrophages as a potential target in ER-positive breast cancer and something that we should think about more, I think, moving forward. Okay, this next study is a study that was published in Nature recently and is extremely interesting but a bit difficult to explain, so I hope I do a decent job of it. The main question that these investigators were asking was, can understanding cell competition actually lead to novel tumor therapies? And so this is a macrophage-focused study again. And what they were really looking at is how cancer cells are competing with the tumor stromal cells, including the macrophages, during the process of tumor growth. And what they did is they developed a mouse model in which they overexpressed MYC along with a MMTV polyoma T. They did that in part because that model expresses high levels of PI3 kinase signaling, and so MYC and PI3 kinase signaling are often found together in breast cancers. And then what they determined was that overexpression of MYC actually led to a competitive, what they called winner state. And this was because when you overexpress MYC in the cancer cells, they express high levels of mTOR, and they can compete with the other cancer cells in the tumor that express less mTOR, but also they can compete with the immune cells in the neighborhood, and that includes the macrophages. Interestingly, if they put these mice on low-protein diets, they could inhibit the mTOR signaling within the cancer cells, but surprisingly, they activated mTOR signaling in the macrophages. And so in the tumor microenvironment, these tumor-associated macrophages, via upregulating actually a couple of key transcription factors. And so what happened in that context was that when you put them in a low-protein diet, the tumor cells actually were outcompeted by the macrophages, and the tumors didn't grow successfully. So what they then did was they showed that if they suppressed activation of those transcription factors and mTOR signaling in the tumor-associated macrophages, they would accelerate tumor growth under the low diet conditions. And alternatively, if they activated mTOR in the TAMs, they could decrease tumor growth under normal conditions. So by reprogramming the macrophages, they could actually decrease tumor growth. So the mechanisms related to this were actually very well worked out in this paper, and I encourage you to read it if you get a chance, but they were related to engulfment. Basically, the TAMs can be genetically reprogrammed to outcompete the cancer cells for nutrient acquisition and for mTOR activation via this engulfment. And that by switching the diet or by switching the genetics of mTOR activation, they could activate the macrophages to be more successful at engulfing, get more of the nutrients, and therefore outcompete the cancer cells. So in this study, they identified a novel innate immune tumor suppressive pathway that could be exploited for cancer therapy. Okay, I believe this is the last of the macrophage-related studies, and in this case, we're going to talk specifically about a type of macrophages called microglia in brain metastases. And the main question of this study was whether we can better understand the tumor immune microenvironment in brain mets to leverage immune therapies. And so what Devin Lawson and colleagues did in this study was essentially they wanted to study specifically breast cancer brain mets because they're very lethal, and we know there are no effective treatments currently. While these mets are known to be infiltrated with tumor-associated macrophages, the role of the brain-resident microglia really remained unknown because they were difficult to distinguish from the tumor-associated macrophages. And so what Lawson and colleagues did was use single-cell RNA-seq in genetic and humanized mouse models in order to understand these different populations. They were able, through this study, to identify the microglia specifically and show that they play distinct roles in inflammation and tumor suppression in breast cancer brain metastases. And as shown over on the, I guess it would be the left, when you have microglia in the brain microenvironment, it increases interferon signaling, it increases antigen presentation, it increases the response of cytotoxic T cells, et cetera. Without microglial, this was all lost. And then I'm going to just show that she did an experiment where they specifically knocked out just the microglia in the brain microenvironment through a genetic model and showed that those mice that had microglia missing were actually, they had reduced NK and T cell responses, they had increased metastasis and decreased survival. And importantly, that effect was also dependent on T cells because when she depleted the T cells, while the microglia still had their interferon responses and their secretory responses, they no longer had their antigen presentation responses, and so they weren't fully functional in the absence of the T cells. So they also went on to show that in human tumors, that pro-inflammatory response is conserved in the human microglia, and the markers of the response are actually associated with better prognosis in patients that have breast cancer brain metastases. And so I thought this was an important study because it contrasts with the pro-tumorigenic functions that are often described in the tumor-associated macrophages and may open up new avenues to specifically activating the anti-tumor function of microglia to inhibit metastases. Okay, so we're now going to move on to why is it so difficult to treat metastases with a focus on plasticity. And this figure here in the front is just to remind people, I don't know if this works. Can I get the arrow? Yeah. That, we're often thinking about things on the level of the DNA, are there genetic alterations? But when you're thinking about plasticity of tumor cells, the cells have to move between different states. And particularly as they go through the metastatic cascade, it's pretty important for those cells to be able to deal with stressors and turn on gene expression in a way that's specific to the context they're in. And so, all these different levels of control are important for gene expression and ultimate survival of cells that are metastatic. And so what I'm going to focus on mostly is epigenetic control as well as translational control of tumor cell plasticity. And what learning about that might tell us about how to deal with metastases. So in this first study, this was a study that was really focusing on MAF amplification. And so the main question of the study was, how does MAF amplification increase metastasis risk? So this is a genetic alteration, and about 20% of breast cancer patients have increased MAF, and that increased MAF through gene amplification is linked to bone metastases. Interestingly, those patients that have the increased MAF also don't respond very well to bisphosphonates. So when they're treated, they in fact sometimes have a counter response or an almost worse response to the bisphosphonates. And so this group wanted to understand why, what happens when you have MAF amplification that makes the bone mets more prevalent and makes the response so much worse. So what they showed by integrating proteomics, transcriptomics, epigenomics, and chromatin accessibility, as well as functional assays, it was a pretty complete study from both human and syngeneic breast cancer models, is that MAF is actually interacting with ER alpha directly. So when you get increased amounts of MAF, it binds to ER alpha and actually reprograms ER alpha, so it binds at additional areas across the genome and now activates the transcription of genes that it normally wasn't activating that facilitate metastatic progression. And so it's changing essentially the chromatin landscape by binding to ER. What they also found is that in addition to interacting with ER, MAF is actually interacting with KDM1A, which is a lysine demethylase that's required for chromatin alterations. And it was shown that the KDM1A was critical for chromatin remodeling in this context. So furthermore, they showed that loss of KDM1A can inhibit metastasis. So in this model, and I didn't explain this very fully, but essentially when you turn on MAF, you get this huge induction of metastasis, and when you inhibit KDM1A with a specific inhibitor, you can reduce the metastasis substantially. So in some ways, the study is, you look at it and think, oh my God, how are we ever going to solve this problem? Because the problem of metastasis involves genetic alterations, epigenetic alterations, and hormonal signaling on top of that. But on the other hand, what was, I think, so heartening about this study was the fact that you could actually take all that information and then target an epigenetic modifier, and by doing that, inhibit breast cancer metastasis. Another study I want to highlight that deals with plasticity is a study around translational plasticity. And so again, I think for many years, everybody's been thinking on the level of gene amplification and then epigenetic alterations and transcriptional changes and how that regulates metastasis. But more recently, we started to realize that translation is not so straightforward and simple. It's not just there's one translation mechanism for all our proteins, but instead, translation is plastic too. And under different conditions, you have different translational functions or molecules that perform different functions. So the main question in this study performed by Bob Schneider was when canonical translation is decreased in response to stress, and remember you're going to have a lot of stressors throughout the metastatic cascade, how do cells, when they're shutting down canonical translation, which is what happens when cells get stressed, how do they still translate genes that are important for migration, or mRNAs, I should say, important for migration, invasion, EMT, et cetera? So how do they stay alive under those conditions? And so in this study, they used genome-wide transcriptomic and translatomic studies to define an alternative mechanism of translation that occurs in response to stress during the metastatic cascade. And so what they showed, if I can get this pointer to work, is that in the primary tumor, you have a mode of typical sort of canonical translation occurring through an EIF4-mediated mechanism that we all know a lot about, I think. But what happens as the cells begin to stress is that canonical mechanism of translation is shut off, and instead a mechanism of translation that's dependent on that's dependent on EIF3D bound to a protein called DAP5. It's canonical cap dependent, so it still binds the cap, but it's a completely different mechanism of translation. And this occurs only when canonical translation is repressed in response to stresses. So what they show is that DAP5 and EIF-D together regulate the translation of mRNAs that are EMT transcription factors, matrix metalloproteinases, integrins, cell survival genes, et cetera, that are critical for cells to survive the metastatic cascade. Let's see. And so here what they did then is they knocked down DAP5, and they showed that while DAP5 doesn't affect primary tumor growth at all, where your canonical translation is mediated by EIF4, it actually does significantly affect metastasis, and hopefully you can appreciate that there. So it's very specific to metastasis. They also went back to human tumors and looked and could show that DAP5 was overexpressed in breast cancer that correlates with poor prognosis. So again, what this underscores is that there's significant translational plasticity also, and that different contexts utilize different translational mechanisms in order for tumor cells to respond to stressors and then be able to metastasize. And if we think more about how to deal with this and learn more about this, we might be able to find specific ways to inhibit stress-induced translation, for example, and inhibit the ability of tumor cells to metastasize. Okay, so the last study I'm going to talk about in the space of translation is a really interesting study that was actually published at the tail end of last year, so in December of 2022, but I just thought it was too interesting to not include still into this year. And that was a study published by Dr. Chavez's lab, in which he looked at tRNAs and how tRNAs regulate metastasis. So the question he really had is, do metastatic cancer cells have altered regulation of tRNA genes? And if so, do they contribute to the disease? And so, as most of us know, humans have 61 codons for 20 amino acids. So there's degeneracy in our codons, and we see, as a result, we have isoacceptors, sets of tRNAs that decode synonymous codons for a given amino acid. So for example, if you look down here, we have two different tRNAs that can put in an isoleucine during translation, I can't read actually from here what they are. It's GAU, I think, and AUA, if I remember. But in any case, they both encode isoleucine. So what he showed happens is that cancer cells surprisingly regulate these two isoacceptors differentially and in opposing manners as they become metastatic. So as the cells are non-metastatic, they express this isoacceptor at very high levels, but as they become metastatic, the second isoacceptor gets overexpressed. Both of them are putting the same amino acid into the protein. They're leading to the same ultimate translation coding. So what's interesting about that is they showed in a number of different models, including breast cancer models, that if you overexpress the isoacceptor, that's the, well, if I could read this, the UAU, you actually get more METs. But if you knock down the isoacceptor, that's the GAU, you also get more METs. So what that tells you is, again, they're functionally opposing each other. And he did this in every direction, actually. So the UAU isoacceptor promotes metastasis, where the GAU inhibits it. The two isoacceptors were found to have opposing effects on the actually translation of growth-promoting and metastasis-promoting genes. So if you go back to this slide, what you see is that when this isoacceptor is used, the codons that encode for proteins that are metastasis-associated growth, et cetera, contain more of this AUA sequence, and therefore, when they switch, you get more of these proteins translated, and therefore more metastasis. They also went on to show that stress, like I talked about in the previous example, actually is critical for changes in these isoacceptor isoforms. And I think really this study highlights a really novel finding that degeneracy of genetic code is exploited in cancer, using these isoacceptor shifts to promote growth and metastasis. And then something I think we haven't thought about a lot, but we have to think about plasticity on the level of translation at many different levels. Okay, so in the last few minutes I have, I'm just going to talk about some really valuable resources I think that will be helpful for many people in the breast cancer community. The first resource I want to mention is from Dr. Sebra's group that was published recently. That's a breast cancer single cell atlas. And what this is an atlas of all the sort of commonly used breast cancer cell lines that we're all using in the laboratory, where they did single-cell RNA-seq to better dissect and understand these cell lines. And the idea behind this is if we really understood these cell lines in all details, we might be able to pick better cell lines that are more reflective of the human disease. And so they did single-cell RNA-seq for 13 breast cancer cell lines. Again, these are the typically used lines in the lab. And they compared these lines to human single-cell seq from 26 breast tumors. And because I'm running a little short on time, I'm not going to talk about specifically a lot of their findings, but I will say what they were able to do with these lines was show that they could come up with novel drugs, for example, that might inhibit a specific ER-positive line dependent on its single cell and the heterogeneity within the cancer cell line. So this will be a really good resource for comparing cell lines to patient samples, but I will say that caveats exist. I mean, I think everybody who's worked with cell lines a lot knows that this MCF7 line that you have in your lab might be nothing like the MCF7 line that somebody has in another lab in another institution. So that heterogeneity may arise from different times of culturing, et cetera, and passaging, so there could still be differences. And I just told you that translational control is important, so knowing about the mRNA maybe isn't enough, but I think it's still a really good starting point for us all to be using this as a go-to resource. Another go-to resource that was published this past year is by Dr. Navin's group, and that was published recently in Nature, and that's a spatially resolved single-cell genomic atlas of the adult human breast. And so what Dr. Navin's group was trying to do was fill a knowledge gap about understanding the non-epithelial compartment of breast cancer, which hadn't really been done in a lot of detail. And so they used both single-cell RNA-seq as well as SN, or single nuclear RNA-seq. And they had to do that in order to capture the adipocytes also. And they profiled breast cancers from 126 women. And really they captured a lot of different cell types, which are shown sort of in the bottom of the slide, but also a lot of different biological states. And so there were a lot of things that came out of this that were interesting. For example, there was an unexpectedly high immune population in the normal human breast that we might have not anticipated. But most importantly, they showed that there are a lot of differences from ethnicity, from age, from menopause, from time, et cetera. And it really underscores the need that when we're all working on breast cancer research and we're doing single cell and we're wanting to compare it back to a normal data set, that we compare it back to an age-specific or a similar data set that's better matching. Okay, it looks like I have 40 seconds left, so I'm going to go through these really quickly. There are resources, so hopefully people can look them up, and I do have the PMIDs on all of these. So another resource I quickly wanted to talk about was a novel model that's just been developed for triple-negative breast cancer in an immune-competent setting. This was developed by Rosalie Sears' lab, and without going into all the details, I'll just say that it's a MYC-amplified P10 deletion model, but what's really nice about this model is that they've taken the time to do all of the single-cell-seq and the spatial mapping, et cetera, and can show that it really mimics the heterogeneity that we can see in human triple-negative breast cancer. And so this model could serve as a new immune-competent model in which to test the therapeutic implications. Okay, so the last example I have is that of Alana Whelm and her group that actually, if any of you just saw her award lecture, she talked about this quite a bit. It was a really beautiful study that she did originally. And then she's put the protocol to be publicly available for everyone, which I think is fantastic. So this past year, they published in STAR Protocols a detailed and easy-to-follow protocol for the entire breast community to establish PDX organoid models and to use for drug screens. So again, I don't have time to go over all this. There's an original paper that she published in Nature Cancer in 2022 that makes use of this model. But what this really is just a really clear and detailed step-by-step protocol so that we can all follow this PDXO model development and use these models, hopefully, to try to predict therapy response and for informing precision oncology in patients. So this is a really useful model for the entire breast cancer community. Okay, so I'm already over, so I'm just going to quickly summarize, but I hope that what I've told you today underscores the fact that understanding fundamental mechanisms of breast cancer are going to provide more insight into more targeted and less toxic means by which we can inhibit the disease. I hope I've convinced you that continuing to understand cooperation between innate and adaptive immune processes may lead to better immunotherapy successes, and that the key to inhibiting metastasis may come through better understanding of the multiple means of plasticity that enable the tumor cells to navigate all the stresses that are experienced during the metastatic cascade, and that we need to continue to develop and improve resources and models for the entire breast cancer community and work together towards the goal of eliminating the disease. Thank you.
Thank you, Dr. Ford. That was absolutely wonderful. Our next presentation is going to be the Year of Review in Translational Research by Dr. Jennifer Ricker from the University of Colorado.
Good afternoon. You get another Colorado presentation. And so I have no disclosures that are relevant to this talk. And I just wanted to thank all the patients and advocates that are here and all the researchers and clinicians and the San Antonio Breast Cancer Program Planning Committee. And as Heide said, it's really difficult to present this selection of work that's been done in 2023 because there's such a huge body of excellent work. And I wanted to differentiate my translational presentation from the two clinical talks that you'll hear from Anne and Mariana. So I selected translational studies that use both preclinical models and clinical samples that I felt were provocative with a high potential for clinical impact. And please keep in mind that it's, like I said, a daunting task to even scrape the surface of the body of work that's been done in the last year. So here's an outline of the topics that I'll cover today. I'll talk about artificial assisted screening, artificial intelligence assisted screening, and postpartum breast cancer a bit, and predicting response to therapy, and antibody drug conjugates, and new approaches to targeting metastatic disease. I found the studies that are investigating the potential for using artificial intelligence software to assist with screening to be quite contemporary and provocative because of the clinical relationship that we all know between a stage and survival. So as you can see here, let me go back here. So can't use that for a pointer. So you can see the relationship between stage. It really affects the survival depending on whether it's local, regional, or distant. And so this rapidly declines with the spread of the disease. So this really underscores the reason that we want to be able to detect breast cancer earlier and more efficiently. So here I'll talk about two studies and that answer this question of can we detect breast cancer earlier, more effectively, and more efficiently. And one was published in Nature Medicine and the other in Lancet Oncology. They both address this question. And these studies provide evidence as to whether this technology can move towards clinical implication. So AI, this first one addresses AI as an additional reader to standard double reading. And it looked at over 25,000 women that were screened in 3 phases. So AI flagged cases for review that double readers marked as no recall, and cases that AI flagged as positive, but the humans marked as negative, were reviewed by a third human. So the AI assistance led to 13% increase in the rate of early detection of invasive and small tumors, with minimal impact on recall rates. This next study, the Messiah study, looked at over 80,000 women in Sweden that were informed that they were in the study, of course, and they were between the ages of 40 to 80 years, and they were eligible for screening at 4 different sites and randomized in Sweden and randomized to AI-supported screening or standard double reading. So the AI-supported screening resulted in a similar rate of cancer detected without increasing the rate of recall, false positives, or consensus meetings. So the follow-up for ongoing, that will be ongoing for the next 2 years will be to assess interval cancer rate as the primary endpoint, and that's quite important. So I actually predict that with continued training, the artificial intelligence assisted screening will become widely used in the next 5 years. And when we look back on it, we'll think that, our kids think that the internet has made this jump from us using the Dewey Decimal System to look up books and articles in the past. And this will be a similar transformative technology, I believe, that would be quite useful. So next I'll move to postpartum breast cancer. And my colleagues at the University of Colorado, Dr. Ginger Borges and Tracy Lyons, as well as their collaborator, Pepper Shadeen, now at OHSU, have really set the stage with these publications that you see down at the bottom for looking into postpartum breast cancer. And as you can see here, if you look at pregnancy and childbirth, there's an increased transient early risk for breast cancer. And then there's a crossover to long-term protection for the young mothers. However, for older mothers, they never see that benefit. And then moving on to lactation, no lactation increases the risk for breast cancer, whereas prolonged lactation decreases the risk. And as in regard to involution, metastasis and death from breast cancer, if diagnosed at the postpartum decade, lead to an increased risk for a poor outcome. So that sort of sets the stage for 2 studies that I'll talk about today. Dr. Amont and colleagues found that poor outcomes in postpartum breast cancer is associated with distinct molecular and immunologic features. So patients that were diagnosed after cessation of breastfeeding have a poorer prognosis compared to those diagnosed during lactation or pregnancy or nulliparous patients, and that the postpartum breast cancer tumors have a distinct molecular and immunologic features. They examined tumor tissues from young women with primary invasive breast cancer diagnosed within 2 years postpartum, during pregnancy, or nulliparous women. So patients with postpartum breast cancers with the low expression of immunoglobulin gene signature and high infiltration of plasma B cells had an increased risk for metastasis and death. Future research will be needed to explore the role of B cell immunobiology in this group of young women that are with breast cancer. And then the next study that I'll present is really important because finding new ways to detect postpartum breast cancer is really a priority, particularly because the high breast density in women of a young age reduces the mammography sensitivity. So here we have a paper that was in Cancer Discovery this year by Christina Sara and colleagues, and you can see that this really is a proof of concept study, and of course more work will have to be done in the future. But this demonstrates that detection of tumor DNA in breast milk is feasible and may be a way to screen for postpartum breast cancer. So they used a unique molecular index, a UMI, a hybrid capture test, and it's mutation agnostic and breast milk-based postpartum breast cancer screening assay. And this is a custom designed panel with 54 genes that are frequently mutated in young women with breast cancer under 45 years of age. So although 53% of breast cancers detected in women that are under 45 years of age are postpartum breast cancers, establishing the benefit of screening in an unselected population will require much larger patient numbers. And indeed, this spotlight by Nick Turner's group underscores that this screening for the ctDNA in breast milk will be most useful in patients that are at high risk because of germline mutations, and those that have had prior breast cancer or prior radiation for other cancers. So a robust comparison between imaging with mammography or MRI with or without the ctDNA in breast milk will be required to establish this, but I think it's really promising and interesting work. So next we'll move on to predicting response to therapy. And here, this is a study looking at molecular profiling of aromatase inhibitors sensitive and resistant estrogen receptor positive, HER2 negative postmenopausal breast cancers. And this was by Mitch Dowsett's group. And here they identified features that are associated with intrinsic resistance that impact the response of ER-positive, HER2-negative breast cancer to aromatase inhibitors, not artificial intelligence. So the promising, so here they look at tumors, 177 of the poorest responding tumors as defined by Ki-67 change after just 2 weeks of neoadjuvant aromatase inhibitor therapy. And that was compared to the good responders, 190 tumors these were all in the POETIC trial. And these were matched for baseline Ki-67. So we know that aromatase inhibitors reduce recurrence and mortality in postmenopausal patients with ER-positive breast cancer, but about over 20% of patients will experience relapse. So these results are quite interesting. They subdivided, let me see if I can find the pointer. They subdivide the more poorly responding tumors into ESR low or ESR1 high. And some of the results are that the poorly responding tumors with the low ESR1 levels were associated with poor response, high proliferation, and high rate of growth or high growth factor pathways and non-luminal subtypes. And the poorly responding tumors with high ESR1 have a lower estrogen response gene profile and a higher levels of tumor infiltrating lymphocytes and more p53 mutations. And also their plasma estradiol levels were lower in these patients. So overall, the immune-related factors are inversely correlated with ER response, and the IL-6 STAT3 pathway was associated with endocrine resistance. So in addition, the poorly responding tumors with ESR1 high had a G2M checkpoint and mitotic spindle hallmarks, and that's evidence of dysregulated control of proliferation. And these patients that show these features, their tumors show these features, they might be good candidates, of course, for CDK4/6 inhibitors. So association of the immune features with poor response to aromatase inhibitors contrasts with the better outcome of patients with triple negative that have high TIL levels. So next, I'll discuss a paper that examines triple-negative breast cancer immunotherapy because it's, of course, demonstrated some benefit, but biomarkers to predict its response to the therapy are needed. And so Ali and colleagues examined spatial predictors, and the multicellular spatial organization and the response to immunotherapy in tumors from patients with TNBC in the NeoTRIP trial is a randomized trial of neoadjuvant chemotherapy alone versus anti-PD-L1 therapy. And the cellular composition and spatial organization of 43 proteins in the triple negatives at baseline, on treatment, and post-treatment found that CD8 positive T cells and the MHC class II on the cancer cells were predictors of response, followed by cancer cell interactions with B cells and granzyme B positive T cells. The responsive tumors contained abundant granzyme B positive T cells, while resistant tumors contained CD15 positive cancer cells, although it still remains to be determined how these cells lead to resistance. So this study highlights that the best predictive value used both the pre and on treatment time points, and of course this highlights the importance of longitudinal biopsies in neoadjuvant studies. So the next study that I'll talk about is on circulating tumor DNA, and this analysis that can improve early stage breast cancer treatment via non-invasive tumor burden assessment and help understand when to de-escalate therapy to limit toxicity for patients who have whose tumors achieve a pathological complete response early, and also potentially to know when to treat, to change up the therapy. So here in this study, we're answering the question of whether ctDNA for monitoring can help with early prediction of response. So serial ctDNA analysis was performed for ER-positive, HER2-negative breast cancer and triple-negative patients receiving neoadjuvant chemotherapy in the I-SPY2 trial. ctDNA positivity rates of ctDNA during and after neoadjuvant chemotherapy were higher in patients with TNBC than in ER-positive, HER2-negative patients, not surprisingly. Early clearance of the ctDNA weeks after treatment initiation, just 3 weeks after treatment initiation, predicts for favorable response to neoadjuvant chemotherapy, but only in the TNBC patients. The ctDNA positivity associates with a reduced distant-free recurrence survival in both subtypes. So gene expression analysis revealed that pathways that were associated with ctDNA shedding at baseline in both subtypes, including active metabolism, proliferation, and high indicators of immune cell activity. And based on these findings, the I-SPY trial will, the I-SPY 2 will prospectively test the ctDNA for utility in redirecting therapy to improve responses. Of course, they acknowledge limitations in the sensitivity and specificity of ctDNA assays. And for example, some patients with residual cancer tested negative for ctDNA after neoadjuvant chemotherapy. And a ctDNA test alone may not provide reliable information regarding disease status and patient outcomes. Thus, improvements in assay sensitivity are needed, and combining ctDNA test results with traditional methods will still be standard for quite a while. So I'll move on to talking about antibody-drug conjugates. And these next couple of slides will delve into some of the issues around ADCs. ADCs are, they use the antibodies to deliver cytotoxic payload to cells that express a specific target antigen, and they're highly specific agents with much less toxicity than free agents. And the established targets include HER2 and TROP2, the trophoblast cell surface antigen 2, and emerging targets with activity in breast cancer include HER3, Nectin-4, LIV-1, B7H4, and AXL. Challenges around ADCs include a lack of studies using combinations of different ADCs and ADCs with other agents. Limited choice of toxins, you can see that it's not a huge variety right now, and resistance to payload toxin due to a variety of mechanisms that I'll discuss in a second, and heterogeneity of antigen expression on tumor cells. And I'll look at a particular study that highlights that. So antibody-targeted release, this review really summarized the mechanism of action of ADCs and also the mechanisms of resistance. And this just came out this year. And I highly suggest reading this review. It was excellent. So antibody-targeted release of the ADC payload causes dying tumor cells to release DAMPs that stimulate dendritic cells to uptake antigen and an increase in MHC class II. And then they migrate to lymph nodes. In the lymph nodes, the ADCs cause a reduction in Tregs and increase MHC class I expression, promoting activity of cytotoxic T cells. They also enhance leukocyte infiltration and expand T helper and effector T cells, as well as natural killer cells and pro-inflammatory chemoattractants, such as gamma interferon. This review highlights ways that the antibody component of ADCs activate the immune system, but I don't have time to go into those now. It also discusses mechanisms of resistance, which can be classified into two categories. Let's see. Yeah, here we go. So the two categories are seen here, and these can be broadly classified as either antibody resistance on the top or payload resistance on the bottom. And again, we won't go into detail here, but we'll discuss in this paper here by Fabrice Andre's group, we'll discuss how antigen presentation, the degree of heterogeneity of expression of HER2 can matter. So basically, in the phase 2 DAISY trial, it looked at the efficacy of trastuzumab deruxtecan, TDXD, in patients with tumors where the HER2 expressing level was either high, medium, or low, or essentially negative. And you can see here that the results that are shown in the waterfall plots, where partial and complete responses are in blue and green respectively, and progressive disease is in red and stable disease is in orange. You can see that while an anti-tumor response was highest in the HER2 expressing high tumors, modest anti-tumor activity was also observed in the medium and low to no expressing HER2 as detected by IHC. So this suggests that a very low HER2 could allow uptake of the TOP1 inhibitor, and that drug efficiency can be partially mediated by HER2-independent mechanisms. So this may expand the population deriving benefit from this drug, this antibody-drug conjugate, and highlights the importance of the emergence of optimized HER2 assays, particularly using immunofluorescence, which is quite a bit more sensitive. Okay. So next we'll move on to new approaches to targeting metastatic disease. So in this study, I highlight a novel way to potentially target metastases. So this study looked at relapse and survival in a cohort of over 1,000 women with non-metastatic triple-negative, and found that use of beta blockers was associated with reduced risk of metastatic relapse, specifically in patients that were treated with anthracycline-containing chemotherapy. And I thought this was quite interesting. In the preclinical models of TNBC patient biopsies, anthracyclines upregulated the beta-2 adrenergic receptor and increased innervation. And in mouse TNBC xenograft models, administration of a beta blocker or genetic deletion of the beta-2 adrenergic receptor delayed the onset of metastases and reduced growth of established metastases. And you can see up at the top the difference in expression of the beta-2 adrenergic receptor is quite distinct. And it actually increases with the anthracycline chemotherapy. So supplementing anthracycline chemotherapy with a beta-2 adrenergic antagonist is a potential therapeutic strategy for enhancing clinical management of TNBC. And in fact, in light of this other study that came out this year showing that subsequent female breast cancer risk is associated with anthracycline chemotherapy for childhood cancers, I think this is a really, since people can be on beta blockers for quite a while, this is a quite interesting potential application of this new discovery. So the next study that I wanted to talk about is how to study leptomeningeal metastasis, or breast cancer LM, so BCLM. And here, tumor cells grow along the lining of the brain and the spinal cord, and it's an extremely challenging form of the disease clinically, and is very difficult to study because accessing tumor material is quite difficult. And so this study used cerebral spinal fluid, cell-free DNA, and CSF disseminated tumor cells, DTCs, to explore clonal evolution. And what they found is somatic mutations with potential therapeutic action were detected in 81% of the BCLM cases, with 19% detectable in the cerebral spinal fluid ctDNA only. So BCLM was enriched in genomic aberrations in genes encoding cell adhesion and cytoskeleton genes, and they also revealed a lobular-like breast cancer phenotype, including loss of E-cadherin, or mutations in E-cadherin, beta-catenin, and the ARGF10 mutations. And this could highlight potential dependencies of this form of the disease. So DTCs from cerebral spinal fluid were cultured in 3D to establish the BCLM patient-derived organoids, which will be a really useful tool for other researchers to study this form of the disease. So I think we're getting close to the end here. In this kind of unique study, Parita and colleagues at UT Southwestern found that fragmented mitochondria in latent breast cancer metastases to the brain enables fatty acid oxidation to sustain redox homeostasis. And fragmented mitochondria in latent brain metastatic breast cancer, like I said, it enables fatty acid oxidation, and depleting the mitochondrial fission protein DARP1, which is involved in the ability of this mitochondria to multiply basically by fission, resulted in increased lipid droplet accumulation and impaired fatty acid oxidation and altered metastasis. Pharmacological inhibition of DARP1 is in development, and they here used a small molecule brain-permeable inhibitor of DARP1 to attenuate metastatic burden in preclinical models. So as I said, these drugs are in development and may someday reach the clinic. And you can see there on the top with the immunohistochemistry picture that the primary tumor is on the top row and the brain metastases are on the bottom row. And you can see how differentially expressed the DARP1, this fission protein, is in these matched primary versus brain met samples. So that's the end of my presentation today, and in short, I just wanted to say that we've made an enormous progress in 2023 towards stopping breast cancer. And I started coming to this meeting in the late 90s, and back then it was nearly all about chemotherapy, and now it's primarily targeted therapy that we're talking about, or how to use the antibody drug conjugates to deliver targeted or chemo to specifically to the tumor cells, so we have fewer side effects. And of course, we're all talking about immunotherapy in a lot of different ways to modulate the immune system. So unfortunately, there's a lot that still has to be done to help patients, and so I look forward to hearing the next person give this presentation next year. See you then.
Thank you so much, Dr. Gregor, for this wonderful presentation. Next, we were going to have our adjuvant early breast cancer year in review by Dr. Ann Partridge from the Dana-Farber Cancer Institute.
Thank you, Virginia. So I'll give the year in review, early breast cancer. And for some reason, I can't forward this. Oh, this guy? Okay, these are my disclosures. And I've got three major themes that I think came out of 2023 in the early breast cancer space. The first is tailoring treatment works, whether that's de-escalation or escalating when appropriate and necessary. The second is that unconventional study designs and real-world data can provide practice-informing and sometimes practice-changing findings, and you'll see some examples of that today. And finally, there's an increasing focus on the whole patient care. We've always aspired to do this, our patients want it, and I think we're getting there, and we should all be proud of that and continue to push in that direction. So let's get started. I'm gonna go along the care continuum, and we'll start with a little bit on prevention, because it all starts there. So we've known for many years that we can prevent breast cancer pharmacologically. We've got tamoxifen, and we have aromatase inhibitors in particular, and other drugs. The IBIS-2 data presented a few years ago showed that we can clearly decrease breast cancer incidence with anastrozole. And you can see the risk reduction, it's a hazard rate of about 51%, 51. And so we also know, and we've known for many decades, that the higher the estradiol in a woman who's postmenopausal body, the higher the risk of breast cancer. You can see on that plot, it's a dose-response curve. And so can we capitalize on that? Well, 2 days ago, in the Wire lecture, Jack Kuzek gave an elegant talk on his work and his career. And one of the things that he showed is recent data that was published in The Lancet this week, yesterday, which showed that baseline estradiol levels can impact on the actual efficacy of anastrozole, and likely all aromatase inhibitors, and their prevention effect. You can see here in the right-sided column that the relative benefit of anastrozole was greatest the higher quartile the estradiol was at baseline in that postmenopausal woman. So you can see that in quartile 1, which is across the top, that's not a hazard ratio, that's a relative benefit. It was about an 18% benefit, and it crossed the 1 confidence interval, whereas when you went up into the other quartiles, there was a higher, much higher benefit, that 50% benefit or so in terms of risk reduction from breast cancer from the aromatase inhibitor. This is exciting new data and suggests that could we potentially tailor prevention by this biomarker? An oldie but a goodie, estradiol, like we treat cholesterol using LDL. Could we dose adjust to the biomarker to get minimally effective dosing and minimize toxicity? Could we do this for patients with hormone receptor invasive disease ultimately and get to that minimally effective dosing? Wouldn't that be great? And I think these are exciting data, and I appreciate the work of Dr. Kuzik and colleagues in this space over the decades. Now let's move on to local and systemic therapy. We'll start with local, and we'll start with local therapy. The Senamac trial was a really important trial, and the question of Senamac was to dissect or to not dissect after a positive sentinel lymph node without any neoadjuvant therapy. So this is pure adjuvant. And this was presented at this meeting, and this was in light of prior studies, including Z11 and Amoros, where there were still uncertainties. There were uncertainties about statistical power, patient selection, radiotherapy fields, and relevant subgroups, some of which you can see listed here. So the investigators tried to overcome this. You can see the eligibility criteria, and they allowed up to 2 sentinel lymph node macrometastases, and patients had to be clinically node negative on palpation, and there was a mandatory preoperative axillary ultrasound. So it's important to know that this was a rigorously conducted trial, very careful, but more inclusive. And you can see here that there were about 36% of patients who had a mastectomy. Adjuvant radiation therapy was given to the vast majority of patients in both arms, those who got the completion dissection and those who didn't have a completion dissection. And most patients got systemic therapy. So it's a treated group of patients from a local standpoint, from radiation overall and from systemic therapy. But it was, do you dissect the axilla or not with low volume sentinel node? And what they showed very clearly was that recurrence-free survival was non-inferior. Thus, doing a completion dissection after low volume sentinel node involvement in the adjuvant setting, again, no neoadjuvant therapy here, did not improve. It was non-inferior to not doing that dissection. So I think these are practice confirming and practice changing for some who were not quite there yet. And certainly with the inclusion of larger groups of patients that weren't studied in the prior work, I think we can generalize this in general, but I'll let my surgical colleagues discuss this further in the trenches, or from the trenches discussion this afternoon. Moving on, we are in the axilla again, where I tread very cautiously as a medical oncologist. And here we have B51, again presented at this meeting by Dr. Mamanos. And what this did was to look and see whether or not we need regional nodal irradiation after a patient goes from node positive, just N1, but pathologically proven, to N0 with neoadjuvant therapy. They enrolled over 1,500 patients, and they randomized them to regional nodal irradiation or not, and with a median follow-up of nearly 5 years, they presented their data and showed very clearly that it is reasonable in the patients who were eligible for this trial to omit regional nodal irradiation after neoadjuvant therapy when the sentinel node goes from pathologically positive to pathologically negative. You can see here invasive breast cancer recurrence-free interval and distant recurrence-free intervals. There is no daylight between those curves. So very exciting data, and I would argue that this too is practice changing or confirming for some who had already adopted this. All right, and then another local therapy study, and this is one of those real-world trials. So this was a study where the investigators had cohorts sent in from 62 centers in 18 countries, trying to amass a cohort of patients where sentinel nodes showed ITCs, isolated tumor cells, after neoadjuvant chemotherapy. And the question, and it comes up in our tumor boards all the time, do you need to do a completion dissection? And the standard has been to do it for most patients, although we hold our noses and don't really want the surgeons to do it, and they don't want to do it, but we needed some data. So they were able to bring together 694 patients with T1 to 4 tumors and 0 to 3 breast cancers, and they found ITCs on 583 cases on sentinel node biopsy. They excluded some. And this is not randomized. This is just observational data, and 182 of the patients had gotten axillary nodal dissection after ITCs found on sentinel node, and over 400 had not. And this is just looking at their observational data. And among those who got the dissection, you can see that 70% had negative additional nodes. Among those who had nodes, only 5% were macromets. Moving on and showing their outcomes data, in terms of axillary recurrence, comparing the two groups, again, not randomized, if they were isolated or combined with local and distant recurrence, it only occurred in about 4% of patients in both groups. Isolated local or axillary recurrence only occurred in between 1 to 2% in both groups. Both comparisons were not statistically significant. And finally, any invasive recurrence, the 5-year rate of invasive recurrence between the two groups was statistically similar. So no clear difference, not high rates of finding meaningful axillary involvement, and not a big difference in terms of invasive events in follow-up. And again, our surgeons tell us that this is practice either confirming or changing, and I think this should be one that's, again, hashed out in the trenches. All right, let's move on to systemic therapy and the exciting updates we saw in 2023. ER-positive disease is where we'll start, and I think this, the immune checkpoint inhibition in the preoperative setting for ER-positive disease was some of the most exciting data we saw presented. We saw this first at ESMO, and this is the schema for the Keynote 756 study, which was presented by Dr. Cardoza at ESMO. It's basically Keynote 522, except now we're giving it in ER-positive disease neoadjuvantly. This is high-risk disease, by the way. Patients had to have high-grade disease, and they had to have enough disease to warrant this kind of aggressive approach. Checkmate 7FL was designed very similarly, just a different IO, so they're using Nivo and a little bit of a different background, and they also, chemo backbone, and they also are selecting patients for, highish grade, and they had stratification around PD-L1. But they basically are doing the same thing in terms of high-risk disease, all neoadjuvant. And the results of these two studies in the neoadjuvant setting in terms of PCR are strikingly consistent, you can see here. So there was a statistically significant difference in PCR with the addition of the checkpoint inhibitor in the preoperative setting to a chemotherapy backbone in these two studies. And very striking and very statistically significant. You'll note that this is ER-positive disease, so these are nothing like what we tend to see in the ER-negative disease in terms of PCR. At this meeting, we saw a very nice biomarker analyses in these two studies, and I'll start with Keynote. And I appreciate Laura Huppard's beautiful presentation yesterday where she discussed these papers, so thank you for the slides. And you can see that on top, the clinical characteristics, really it was just lymph node involvement. There was a little less benefit in the patients who were lymph node negative, as you might expect. But focusing on the biomarkers, you can see that there's almost a dose response curve in terms of the greater the PD-L1 positivity, the higher the likelihood of PCR. Remember, this is ER positive disease, so if you look on the top right column, if you have a CPS of 20 or greater, those patients, again, small sample in general, but it's over 200 patients, had a PCR rate of 53% when the checkpoint inhibitor was added. Of course, the patients who did not get the checkpoint inhibitor also had a higher likelihood of PCR when they had PD-L1 positivity that was higher. ER status, we saw not unexpectedly that the lower the ER status, the higher the likelihood of PCR as well. In Checkmate, this was also extremely elegantly done by Dr. Loy and presented and showed that, again, that the dose response curve that you can see in terms of PD-L1 status and the likelihood of PCR, ER status similarly, the lower the ER status, the higher the likelihood of PCR, and particularly when the checkpoint inhibitor was added. TILs also added additional both prognostic and predictive value in this setting. Interestingly, Ki-67 had no association with PCR. So what do we make of all this at this point in time? I think these are very robust, exciting, and provocative data, but we have to wait for the event-free survival data. Now we do have some biomarkers, but we need to determine the best biomarkers or combinations to predict not just PCR, but ultimately event-free survival, because of course the toxicity is not trivial with these drugs. All right, moving on in the ER positive space, I think one of the other most highly awaited data, for years we've been waiting on NATALY, and here you can see the schema for the NATALY trial adding the adjuvant CDK4/6 inhibition ribociclib to endocrine therapy in the adjuvant setting. And we got an update on this. We heard it at ASCO. We got an update on this from Dr. Hortabaji at this meeting. He showed that there was early discontinuation in about a third of patients, but they were further along than they were in June when they presented it. So now a larger proportion of the group has completed the 3 years of ribociclib in addition to the aromatase inhibition and the control group's comparative. And what NATALY showed in the updated analysis is that the median follow-up for invasive disease-free survival at 33.3 months, and the absolute IDFS benefit with ribo is about 3%. This is a reduction of 25% in terms of invasive disease with ribo added to AI. Across key subgroups, there was clear benefit, and again, we're having a hard time finding biomarkers in the CDK4/6 inhibitor work. The distant disease-free survival was similarly impressive with a 2.7% difference at 3 years. So small, but remember this is a lower risk group. There were node negative patients in NATALY. The overall survival data is too immature, and there are not enough events yet, so we'll wait for that, but it's going to be a long time, we hope. And just to put this in context, the monarchE data was also updated this year at ESMO, and you can see the schema here. It's 2 years of abemaciclib in a high-risk population, generally higher risk than what was included in NATALY. And you can see that the benefits from monarchE showed sustained improvements at 5 years for both invasive and distant relapse-free survival. Here's the distant on the bottom. The the absolute benefit at the 5-year mark is about 7.5% in terms of invasive disease-free survival events. So very impressive. So what do we make of these data at this point in time? I think both ribo and abema improve event-free survival in high-risk early breast cancer that's hormone receptor positive. And I didn't show these data, but half of the events in the abema data are distant events, and we're markedly reducing that. All subgroups seem to benefit, including those by Ki67, ER, PR. So we really do need some biomarkers. A few hours ago, Nick Turner presented some data suggesting that maybe MYC amplification is associated with resistance to CDK4/6 inhibitors, so we'll have to get more data on that. Certainly, adjuvant abema is currently approved, and we're using it for our high-risk patients, and we await the approval of ribociclib and expect that it may be in light of the robustness of the data. But there are many other unanswered questions. Can we use the CDK4/6 inhibitors to replace chemotherapy in the adjuvant setting, not just add to it? And what about adherence and toxicity in the real world? We know these are issues with hormonal therapy already, and I showed you the adherence, the dropout rate in terms of NATALEE. It's also high in abema, not quite as high, but NATALEE goes on for 3 years. And so I think we need to do some real-world work around this and obviously continue to support our patients as much as we can with toxicity management. All right, let's move on to triple-negative breast cancer. And what's the new hot data? Well, there's KEYNOTE. And KEYNOTE we're all familiar with, 522, which randomized patients to receive pembro or not with a carbotaxol AC backbone. And we got updated data at ESMO, and we just had updated data a few hours ago here. These are the ESMO curves. I don't know how much they changed because it was, I think, noon when those things were being presented by Peter Schmidt. But I don't think this changed very much. The absolute difference in terms of 5-year event-free survival on the left remains quite robust at 9% for preoperative, the addition of pembro to preoperative chemotherapy in triple-negative disease. And you can see the event-free survival by PCR status. Now, this is really exciting. Look at those curves and among the patients who have the PCR. It begs the question, do they really need the adjuvant pembro if you got a PCR to chemo and you do that well? And we are, of course, studying that in the optimized PCR trial run through the Alliance. But I also want you to focus on the lower curves. Those are the no PCR curves. Pembro helped those patients too. That's the green line at 62% if you got pembro even without a PCR. But that's still And then those patients who didn't get the pembro, obviously they won't hopefully exist anymore. We won't do that anymore. But for the patients that really need it and they get the pembro, we still see that there is work to be done. And there are a number of studies that are enrolling right now, including the optimized RD trial, which is open and enrolling and randomizing patients who get a KEYNOTE regimen, don't have a PCR, and get randomized to receive sacituzumab or not, in addition to the pembro with or without capecitabine. That's investigator's choice. You can see here in KEYNOTE 522, distant relapse-free survival, again, very impressive, and then that's at 7.8% at the most recent data cut, 5 years. We await overall survival data, and my gut is we're gonna see it. Just a few hours ago, Dr. Schmidt presented these data. Many questions come up, how low do you go? And he showed that in stage 2 disease, we still see benefit from the pembro. That's pretty significant. In node-negative disease, we see some pretty significant benefit. And then finally, in the 2 and 0s, there's a 10% absolute difference in terms of event-free survival from the addition of pembrolizumab. So very impressive, approved, and available in many places, and we hope more so moving forward. As we get excited about these therapies, we need to remember that there is serious toxicity from them, and we're seeing it more and more. This is a shout out to the real-world toxicity paper that was presented here. I think it was in one of those poster spotlights or a mini oral, and this was a multi-center retrospective cohort of patients who received just standard of care keynote outside of a trial. They got it from 17 sites. They used medical records among 577 patients, and what they found was an extremely high rate of hospitalizations as well as acute care utilization. More than 30% of patients, whether they were black or white, got hospitalized. This is in the United States, run out of WashU. So we'll see when this manuscript comes out what they need to control for, but I think this is just a cautionary note that we need to continue to monitor these patients carefully, both on therapy and when they come off therapy for these more novel toxicities from immunotherapy. Neotrip was presented at ESMO this year, just for everybody's to reminder. This was a different chemo backbone, but all adding atezolizumab in the neoadjuvant setting. And unfortunately, Neotrip was negative in terms of their outcomes. This is event-free survival. And why is Neotrip negative? Different population, different chemo backbone, it could be the reason. Different drug, atezo is a PD-L1 inhibitor as opposed to pembro, a PD-1 inhibitor. We're waiting on Jeperduze, that's pending, and also using atezo. And another study that was presented at this meeting, the Impassion 030, which randomized patients in the pure adjuvant setting with triple-negative breast cancer to the addition of atezolizumab again or not. And this study met its futility endpoint, and it was ineffective, unfortunately, at a median follow-up of 25 months. So these are the key secondary efficacy endpoints, also clearly a negative trial. So why is Impassion 030 negative? Maybe it's because it's in the adjuvant setting only. Do you need more tumor in there to have those neoantigens to jazz up the immune system? It's a different population, a different chemo backbone, different drug, again, atezo, and we are all waiting on S1418, the SWOG trial, adding pembrolizumab in the pure adjuvant setting to help inform this, because we all know sometimes we have patients who you thought it was a small tumor, and then they go to the OR, and it's either a bigger tumor or lots of lymph node involvement, and we send those ones to our tumor boards. All right. Now, finally, in terms of the systemic therapy, HER2-positive disease, again, a few hours ago, you heard the most recent update from the Catherine study, TDM1 versus trastuzumab in the residual disease setting in HER2-positive breast cancer after neoadjuvant therapy. Dr. Leubel presented these data, and at a median follow-up of 8.4 years, there's an absolute invasive disease-free survival benefit of 13.7% at 7 years. Very, very exciting, very clear win for patients. You can see here the site of first recurrence as an event. Most were distant, and CNS recurrence is still a problem in this population. Here are the overall survival data. There's nearly a 5% benefit at 7 years. This is very exciting, and we've been doing this for our patients, but it's nice to see a survival benefit report out. Let's move on to survivorship and supportive care in my last few minutes. Can we improve patient outcomes with lifestyle interventions? This is something that we've all been interested in. If we could get our patients to take better care of themselves, if we could get ourselves to take better care of ourselves, maybe people would do better. BMI has been a focus, and the obesity epidemic, particularly here in the United States. We know that for breast cancer patients, there's increased breast cancer mortality for patients who have a higher BMI at diagnosis, as well as an increased risk of second cancers. The Be Well trial was designed to try to mitigate this in breast cancer survivors, to help them, if they were overweight or obese, to lose weight, not just tell them to exercise and diet, but a very well-controlled, well-designed health education, telephone-based weight loss intervention was designed versus a health education alone control arm. The BUL trial is fully accrued, and my colleague, Dr. Jennifer Ligabell, presented at FASCO the weight loss data, and the results showed that the patients clearly lost weight, both at 6 months, and then this was sustained in the intervention arm with the BUL intervention at 12 months. You can see here the control group actually gained weight. So there was a significant weight loss in the intervention arm. Now, interestingly, a sister study was conducted in France and reported at ESMO by Inej Vaz-Louise. They only had about 200 patients, but they showed that in a French setting with French patients and French food, they could also do the exact same thing. They also lost weight. And so we know that this is a potentially generalizable intervention, and what we don't know yet is whether it's going to impact event-free survival. But weight loss is a pretty nice intermediate outcome, so I thought I'd present it here today. And good for our patients to know we can do this. So we're excited about that. And stay tuned on the event-free survival. The positive trial was presented here last year, and that's the study where patients are stopping their endocrine therapy for up to 2 years to have a pregnancy and then get back on. These are the results of that paper. And at this meeting, the time to pregnancy data were presented by Hatem Azim, and this showed that age was really the only predictor of time to pregnancy. Women under 35 got pregnant faster. Age matters in this. When we looked at the use of assisted reproductive technology and the chance of pregnancy, you can see that age predicted getting pregnant at all, as did the use of cryopreserved embryos or oocytes that then were made into embryos and transferred. There were no other big predictors of becoming pregnant. We looked at the safety of ovarian stimulation for fertility preservation, and that means before enrollment, at diagnosis, going through the procedure to bank eggs or embryos, and as you can see here in the curves, at 3 years, the breast cancer free events were very similar, whether someone did that or did not. And then we also looked at whether or not a woman used assisted reproduction while on study. And about 80 patients actually got stimulated during their break from therapy. And as you can see here, there isn't any clear signal for harm. I say this cautiously because the numbers were very small, there were very few events, but at least we don't see signal for harm. And when these women are taking their break from their endocrine therapy, they kind of want to do everything they can to get pregnant and get back on. So I think it's reasonable to do this, again, with the caveats of small numbers and not a ton of safety data. All right, so the key takeaways from 2023 and early breast cancer, I would argue, are tailoring treatment, de-escalation or escalation works. We saw this in the breast and the axilla in our surgical colleagues' work. We saw it in terms of the tailoring with regard to, in particular, the data presented by Dr. Loibel for Catherine. Unconventional study designs like positive and real-world data like that ITC study. It's pretty practice-informing or practice-changing. And finally, that focus on research for the whole care of the patient, I think, is really exciting. So thank you very much, and I'd like to thank the organizers for having me for this session, and all of the people who've helped me with this presentation.
Thank you, Anne. I don't think you slept much the last 3 days, but we truly appreciate this review. For the last talk of this session, Dr. Mariana Chavez Gregor from UTMD Anderson Cancer Center will talk to us about metastatic breast cancer.
Hello, everybody. I want to start by thanking the organizer committee for this privilege. These are my disclosures. And I have the challenge of following 3 amazing presentations and summarizing what, in my opinion, has been an extraordinary year of advances and progress in the care of patients with metastatic breast cancer. So what I decided to do was to emphasize what I think are potentially practice-changing findings. This is, of course, a non-exhaustive summary. So I want to start by acknowledging and recognizing numerous teams of investigators, research teams, patients, caregivers, and all of us that are working tirelessly to improve the lives of patients living with metastatic breast cancer. Lastly, another 2 disclosures. This is going to be a little bit of a fast ride, and it is my opinion. So a lot of that, it's my interpretation of very new data, so please consider that while analyzing this talk. So what I've decided to do was to split the talk according to breast cancer subtype, and most of the time we're going to spend talking about hormone receptor-positive breast cancer, where in my opinion we've seen some of the most strong reports or greatest advances this year. We know that CDK inhibitors have become standard treatment in the first-line setting. We have here a number of the studies in the first-line setting that are evaluated the use of CDK inhibitors in combination with AIs. The designs are similar, and as you can see in this table, the results regarding progression-free survival, which was the main outcome of the studies, it's incredibly similar, not only in the absolute numbers, but also in the hazard ratios. What we have been wondering about is the overall survival. And as you can see in the bottom of the table is that there we have some studies that are positive, the ones actually evaluating the use of ribociclib, with the results from the Paloma 2 trial being negative for overall survival. And we've been missing the results from the abemaciclib data that was presented this meeting. Of note, we have data in the first-line setting in the Mona Lisa 3 study, which is a study that compared included patients both in first-line and second-line setting treated with fulvestrant. But we're wondering about the results in the overall survival. And we heard earlier in this meeting the results from the Monarch 3 trial that demonstrate a very significant improvement in overall survival in patients treated with abemaciclib in the first-line setting. While this was not statistically significant, it is a dramatic difference in separation of the curves, and actually it's the greatest absolute difference that we're seeing in all the CDK4/6 inhibitor. Now, it's important to mention that this is a relatively small trial, smaller than the other studies, and that randomization was done 2-to-1. Also, it's important to mention that in a post-discontinuation treatment, there was a 32% of use of CDK inhibitors in patients during the placebo arm and 12% in the abema arm, and these are results at 8-year follow-up. So really, really encouraging results. Now, while CDK4/6 inhibitors are clearly standard of care in patients with metastatic breast cancer, I think we all still have a lot of questions regarding these agents. And of course, we wonder, why are the overall survival results different? And I think that we need to interpret these results remembering that the primary endpoint of all these studies was progression-free survival, but of course, we can ask, why is this related to differences in study conduct and design? You know, there were issues related follow-up in the Paloma 2 study. I talk about differences in the randomization 2-to-1 or smaller sample size in Monarch 3, but it is also possible that in addition, there are drug differences that could be associated with efficacy, in particular overall survival, because we're not seeing any differences in progression-free survival. Now, we also can ask ourselves, what is the best endocrine therapy partner? Should we combine it with aromatase inhibitor or fulvestrant? Do all patients really need to receive these drugs in the frontline setting? Of course, we all are experiencing the challenging decision of what to do at the time of progression in our patients. Is there a role for using CDK inhibitors beyond progression in a previous CDK inhibitor? And what are we going to do with the incorporation of CDK inhibitors in the adjuvant treatment in these patients? And ultimately, what is the role of combinations? And this year, we had answers, at least partially, to some of these questions. Regarding the question of best endocrine therapy partner, the Parsifal trial was updated during this meeting. This trial is an international multi-center phase 2 trial assessing whether fulvestrant or letrozole are an optimal endocrine therapy partner for palbociclib. So in this updated study, we confirmed what we had previously heard, that there's no difference in terms of what endocrine therapy partner we use. And actually, the PFS is exactly the same for combination of palbociclib with letrozole or with fulvestrant. Now, something that I think it's relevant to mention is that there was also no overall survival difference, but I think it's relevant to note that in this study, again, it's small, but the overall survival of both patients treated with palbociclib, either with AI or fulvestrant, reached a number that is very similar to the updated overall survival analysis that we heard from Monarch 3 today, this meeting. Now, well, the benefit of CDK4/6 inhibitors in the first-line setting, I think it's not questionable. I think there's actually room to believe that it's possible that a sequential approach could be optimal in some patients. This was a study conducted by Dutch investigator presented at FASCO this year, a very elegant and truly a sequence design where over 1,000 women in the Netherlands received treatment sequentially. Either they received CDK inhibitor in the first line combined with AI, followed by fulvestrant at time of progression, or AI alone, followed by fulvestrant in combination with a CDK4/6 inhibitor, with primary endpoint being PFS2. Now, it's important to remember that this study included most of the patients who were actually treated with palbociclib as a CDK4/6 inhibitor, 91% of the population in both arms. Only 80% of the patients were treated with ribociclib and 1% with abemaciclib. In this group of patients, 35% of the participants had stage 4 de novo, 56% of the patients had evidence of visceral disease, and half of the participants have a disease-free interval of greater than 24 months. Not surprisingly, in the first-line setting, in the SONIA trial, patients treated with CDK4/6 inhibitor initially had improved progression-free survival, similar with what we have seen consistently in clinical trials. However, the primary endpoint, progression-free survival, demonstrated that there were no differences in the subsequent line of treatment. There were also no differences in overall survival. This was clearly an impactful clinical trial and actually demonstrates that CDK4/6 inhibitors in the first-line setting compared to the second line does not improve progression-free survival, overall survival, and actually patients treated with CDK4/6 inhibitors in the first-line setting, of course, had a more higher risk of side effects. Now, I think these results are very interesting, and while I don't think are changing practice in the United States, I think it makes us comfortable maybe knowing that there's some group of patients that probably can have great outcomes delaying the use of CDK4/6 inhibitors. Of course, one of the themes in the meeting is how to select or identify these patients, and I don't think we know how to do that yet. I think these results are also potentially very relevant for low-resource settings where resource optimization has to be determined, and it's key. So I also mentioned that we have a lot of questions about what to do at the time of progression. So questions remain regarding the optimal next line of therapy in patients' progression to CDK inhibitors. And in ASCO, we heard the results from the Palmyra trial. In this study, 198 patients were randomized to receive palbociclib after they progressed in a palbociclib-based treatment. Patients could have received fulvestrant or letrozole, depending on what they received at initial endocrine therapy, and actually 88% of the patients receive in both arms aromatase inhibitors. So most of the patients receive either fulvestrant in combination with palbo or fulvestrant alone. So this was, again, trying to answer the question, what is the role of continuing palbociclib after progression on palbociclib? Palmyra was a negative study. Palbociclib maintenance following the progression on palbociclib therapy did not improve outcomes compared to endocrine therapy alone. Now, if you see the curves, you can see that at the analysis at 6 months, the curves look that they are separating a little bit, suggesting that maybe there are some patients that could benefit from this strategy, but once again, I don't think we have identified these patients yet. Now, the proportion of patients that receive palbociclib for greater than 12 months was similar in both arms. And as you can see, there's an analysis according to PFS duration on palbociclib. And in patients that receive palbociclib in the first line for greater than 12 months, we see again that maybe we can see a separation at that 6 month. But I think that is hypothesis generating and not necessarily practice changing for many of us. So the results of Palmyra really add to the previously presented data evaluating the use of a CDK inhibitor following a CDK inhibitor. We have 3 phase 2 trials that are listed in this slide. 2 of them are negative, the PACE and the Palmyra trial, and the trial that it's positive, it's a maintained study. What I will highlight is that, well, we cannot compare trials. The maintained trial is one that switched the type of CDK inhibitor in its design. Most patients received palbociclib, and then they were randomized to receive ribociclib or not. What I also think it's very interesting is for us to keep an eye on the control arms. Like these patients, while they can do great on CDK4/6 inhibitors, do not have great outcomes on the next line of therapy. Now, the next big question is combination. And I think that takes us to one of what I consider to be the highlights of our meeting this year, that are the results of the INAVO 120 study. You can see the study design here. So INAVO 120, it's a clinical trial in the first-line setting, patients with metastatic hormone receptor-positive breast cancer, high risk. This study recruited patients that have PIK3CA mutations, but key is that these patients had progression during endocrine therapy or within 12 months of completing adjuvant endocrine therapy, so a very high risk group of patients. And patients were randomized to receive palbociclib and fulvestrant in combination with placebo or the combination with inavolisib. And this new drug, what it is a highly potent selective inhibitor of the alpha subunit of PIK3CA. So this cohort of patients, as I mentioned, were high risk. 40% of them were premenopausal, 80% of them have visceral disease, and 35% of them have progression during the first 2 years of adjuvant treatment. And I think that's a group of patients that we always wonder what's the optimal treatment. The results were truly remarkable, and the primary endpoint was positive. The addition of inavolisib to palbociclib and fulvestrant improved progression-free survival, almost double PFS from 7.3 to 15 months with a hazard ratio of 0.43, with curves that, as you can see, separate very early on. Now, the overall survival results are not mature yet, but we see a trend in the first interim analysis with a hazard ratio of 0.64. So we are all very excited about these results and the implications that they can have for our patients. Now, the combination not only improved progression-free survival, but they also improve overall response rate and duration of response. Now, as we think about a lot of these agents and combination, we of course have to think about the safety in our patients. So this is a list, a table showing the adverse events associated with the use of the combination, and we can see high rates of neutropenia, not surprisingly, likely all driven by the palbociclib. Now, there are known side effects associated with inhibitors on this pathway, and we can see they highlighted here. So as we think about this data, I think it's important to focus it on the population that it targets. And it targets those patients that have likely resistant disease to endocrine therapy, something that is very selective and different than all the previous trials that I've talked about in the first line setting. Now, as we think about endocrine therapy and hormone receptor-positive tumors, it's key to talk about elacestrant, an oral selective estrogen receptor degrader. The results were presented in the Emerald trial that randomized 477 patients previously treated with endocrine therapy, and all of the patients were previously treated with CDK4/6 inhibitors, representing a contemporary cohort. Now, patients were randomized to receive elacestrant, or most of the patients received fulvestrant otherwise, and this was a positive study. These are the main results from Emerald, and I will note 2 things. One, note the dramatic drop in the curves in the first 3 months. Almost half of the patients have progression very early on, talking to us about those already resistant to endocrine therapy patients. It's a positive study in the overall cohort, but as you can see, there's a greater benefit in patients that harbor ESR1 mutations. Now, we learned last year here at San Antonio that amongst patients with ESR1 mutations that had a duration of response with CDK4/6 inhibitors greater than 12 months, the benefit is much larger. Here we're talking about median progression-free survival of over 8 months compared to 1.5 months in the control arm. And remember that majority of the patients in the first-line setting treated with CDK4/6 inhibitors have a median PFS of 24 months. The majority of patients that we're treating will fall likely into this category, talking about an important benefit of elacestrant. Now, I'm bringing this up in this meeting because it was this year where elacestrant became approved by the FDA to treat patients that harbor an ESR1 mutation that have been previously treated with endocrine therapy. Now, in this meeting, we heard very interestingly about the effect of elacestrant in patients that have other coexistent alterations, and I personally think it's relevant to see that independent or other characteristics, particularly PIK3CA mutations, we can still see a benefit on elacestrant in these patients. I personally have patients that have both ESR1 and PIK3CA mutations, and I think we don't know what drug to pick first in these patients. So again, moving on another new and exciting drug, we're going to talk about capivasertib. Capivasertib is a potent selective inhibitor of all 3 isoforms of AKT. And the CAPItello-291 trial, it's a trial that randomized patients previously treated with endocrine therapy to receive capivasertib in combination with fulvestrant or fulvestrant and placebo. In this trial, almost 70% of the patients received previous CDK4/6 inhibitors, and about 18% received chemotherapy previously. This is a positive study. As you can see, in the overall population, there was a benefit of the addition of capivasertib, and in the 44% patients that have altered population, we see a separation of the curves, and actually the benefit in the overall population was felt to be driven by the patients in the altered population. Now, it is important to mention that in subgroup analysis and the altered population show benefit of capivasertib regardless of different characteristics. And while overall survival is not mature yet, early analysis shows separation of the curves. Now, capivasertib in the United States was approved just 3 weeks ago to be used in patients in the altered population. We're talking about not only PIK3CA, but also AKT1 and PTEN, and that is today another option for us and for our patients. As we incorporate this drug in our treatment armamentarium, I think it's key to remember the side effects associated with this drug, and one of them is that we need to be very cautious with diarrhea and treating it aggressively and early on our patients. Despite the side effects, we heard in this meeting results from quality of life studies that showed that overall patients do very well with this regimen, and with the exception of diarrhea, the rest of the symptoms, like patients did very, very well. Also in this meeting, we heard regardless of the specific alteration in CAPItello-291, there was a benefit of the curve of the drug. Now, most of the patients had a PIK3CA mutation. It was a smaller number of patients that had an AKT1 or a PTEN alteration, but we saw a benefit in all of them, giving us, certainty and more data when we incorporate the use of this drug into our practice. Now, in this table, I show the drugs that are inhibiting this pathway. Let's not forget about everolimus that now has been approved for over a decade now, and we also see there listed the results of the SOLAR-1 study that we're using alpelisib in combination with fulvestrant in patients with PIK3CA mutation. And while we cannot compare studies, again, let's look at the control arms in all of these trials, how similar they all are. Now, we don't know how a patient that had progressive disease in one of these drugs will do in another one, but I personally don't think there's going to be a lot of benefit since we're talking about the same pathway. What we should acknowledge is that all these drugs have similar side effects, but in different percentage. So we should take this into account while using this drug in our patients, and remembering that we need to take into account stomatitis, rash, diarrhea, and hyperglycemia is a big one, with actually actually reassuring data on capivasertib that it's way less common and severe compared to alpelisib. So a talk on the year in review for breast cancer will be incomplete if we don't talk about antibody-drug conjugates, and we heard an extraordinary discussion about resistance early on by Jennifer, but let's quickly remember some of the studies that have been updated this year. One of them is Destiny Breast 04 that used trastuzumab deruxtecan, a trastuzumab humanized HER2 monoclonal antibody with a topo-1 payload that was evaluated in the Destiny Breast 04 study in patients with HER2-low tumors. This is a positive study, and we heard at ESMO this year updates on this trial, and with now a median follow-up of 32 months, Destiny Breast 04 continues to show sustained clinical benefit in both progression-free survival and overall survival in our patients, and it clearly continues to solidify itself as a great drug for the use of our patients that have HER2 tumors. Now, the other ADC that we're incorporating into our practice is sacituzumab govitecan. Just to remind you, sacituzumab govitecan is a TROP2 inhibitor, also with a topo-1 payload, and the trial that evaluated its use in hormone receptor-positive patients was the ASCENT trial. In this trial, patients were randomized to receive sacituzumab govitecan or treatment of physician's choice, with primary endpoint being progression-free survival. With the extended follow-up that we heard from Sarah Tolaney in ASCO this year, we see that the use of sacituzumab govitecan is associated with improved progression-free survival and overall survival in this group of patients. We also continue to see that at each one of the landmark analyses, patients treated with sacituzumab govitecan do better. We also heard updates on the benefit in patients that are HER2-0 or HER2-1 or 2+, and very interestingly, the results of the TROP2 expression. And as we can see, there's a benefit in patients that have low TROP2 expression or high TROP2 expression, but I think we're gonna keep hearing more because maybe there's some additional measurement that we can do in TROP2 to identify patients that are gonna benefit for this drug, although that's something that we're not doing at this point in clinical practice. Sacituzumab govitecan was approved by the FDA this year to be used in patients with hormone receptor-positive breast cancer, added to the indication that we had previously from patients in the triple-negative setting. Now, the new antibody that we heard about at ESMO and in this meeting was Dato-DXd. Dato-DXd, it's a TROP2 antibody with deruxtecan. So it's a TROP2 antibody, just like SASI, but with a deruxtecan payload, just like trastuzumab deruxtecan. And this is a phase 3 randomized trial, randomizing previously treated patients to receive Dato-DXd versus physician's choice chemotherapy. Now, all patients should have received endocrine therapy, and previous CDK4/6 inhibitor was not mandated, but 80% of the patients were previously treated with CDK4/6 inhibitors. These are the results from TROPION-1, a positive study, improving progression-free survival by about 2.5 months with a hazard ratio of 0.64, with curves that separate early on, as you can see at the different landmark analysis, a separation of the curves that it's, quite notable. Now, the overall survival results are not mature, but Aditya Bharti updated the results this meeting to see that those patients treated with Dato-DXd had a delay in next subsequent therapy, and that this was similar regardless of the duration of CDK4/6 inhibitor in those patients that received those agents. The response rate was also higher, and as I said, the overall survival results are not mature, and we're gonna have to wait for those results. Now, this drug, it's associated with some interesting side effects, so I think it's important to notice that we're seeing way less neutropenia than what we see with standard chemotherapy, but there was a proportion of patients with dry eye and stomatitis, and while that does not seem to be severe and mostly grade 1, it's something that we will have to remember if this drug becomes to market. Also see at the bottom of this slide the low number of patients with pneumonitis, which is, I think, something that is reassuring now that we're learning more how to follow up these patients. Now, this is a table that summarizes the ADC trials evaluating the results in different in the hormone receptor positive population. Remember that we're talking about similar payloads. They are all like topo-1 inhibitors, but the antibodies are different, and also the studies are different, but we're talking about a similar patient population with similar PFS in the control arm. So I don't want to compare the drugs, we cannot do that, but it's telling us a story about these patients that we need to improve the treatments that we have, because in the placebo arms, on the standard chemotherapy, the PFS, it's around just 4 months. So there are a number of questions remaining in the ADCs for breast cancer in terms of what's the best sequence, how we are going to eventually incorporate them in the first line, and a lot really related about resistance. So there was a lot this meeting regarding how we can really optimize the use and really use these drugs in a better way for our patients. There was an extraordinary session. The discussion was done by Ana Garrido regarding the sequence of ADC by ADC, and well, the results are very different. I think the take-home point is that in all cases, the PFS after this, or for the second ADC, is shorter than for the first one. So what we have to acknowledge is that the data is limited, and we still have a lot of questions, and we're going to have to wait for clinical trials to really address the question of sequence. Today, the best sequence for all the patients, it's really unknown and it's not clear. But what we can tell is that this retrospective data suggests that after ADC1, the PFS using ADC2, it's going to be shorter. Now, what we don't know is how that compares with standard chemotherapy and how to identify the patients that are going to do better or they're going to do worse. And it seems that maybe one of the suggestions for some of these posters is that topo-1 variants are a possible mechanism of resistance. So briefly, how to integrate that in practice. So I personally think that in the first-line setting, endocrine therapy with CDK4/6 inhibitors remains to be the standard care. And at least in my practice, I do not use CDK beyond progression outside of a clinical trial. Now, we cannot have a rule for all our patients, and we need to take into account endocrine therapy response, sensitivity, CDK4/6 duration, but also, it's key to remember the role of somatic mutations and germline mutations, because all this combination are potentially useful for our patients in the second-line setting. I do believe that many of our patients benefit from second-line endocrine therapy-based treatment, most of them in combination. Now, we do not know how many of these patients will do if in the third-line setting we use, for example, elacestrant after olaparib in a patient who has a BRCA1 mutation, or capivasertib after the use of elacestrant. But in some patients, I believe we're going to be able to use not only a second line of endocrine therapy, but also a third in combination. Now, most of us will use chemotherapy after that, because remember, the ADCs got approved after chemotherapy use, and I personally continue using it. Most of these patients keep having, given the quality of life implications that this drug has. Now, after that, it's key to remember that HER2 status determination is going to help us to see if our patients are eligible for trastuzumab deruxtecan or not. Now, whether the new drugs will come to market and how will we position, I think remains to be seen. But I think the combination, even inavolisib, will likely be just for selected group of patients, and probably Dato-DXd will come and compete with sacituzumab govitecan with similar results in the studies and likely a more favorable toxicity profile. Using the few minutes that I have left, in triple-negative breast cancers, we know that we are using now routinely immunotherapy and antibody-drug conjugates. And just very briefly, I want to highlight the results at ESMO this year of the small arm 7 of the Begonia trial, where patients in the first-line setting with triple-negative breast cancer, unselected population, were treated with Dato-DXd in combination with durvalumab. So ADC combined with IO, and In this single-arm study, median PFS was almost 14 months. Again, we're not necessarily comparing trials, but in the population of Keynote 355, PD-L1 positive treated with Pembro, we talk about median PFS of around 9.5 months. So clearly interesting, and now the Begonia arm 8 in the PD-L1 positive population, it's currently recruiting patients. To finalize, I'm gonna just briefly talk on HER2 positive tumors. We know that we have a lot of drugs, extraordinary drugs for treatment of these patients. And we heard in this meeting the results of the HER2 Climb 02 study. HER2 Climb, it's a trial that it's asking the question of what is the efficacy of T-DM1 in combination with tucatinib versus T-DM1 alone? Let's remember briefly, with HER2 Climb initially evaluated combination of tucatinib, capecitabine, trastuzumab versus versus trastuzumab and capecitabine alone in previously treated with pertuzumab and T-DM1 combination, and now we're evaluating newer combinations with tucatinib. About 44% of patients in this trial had brain metastases. And this is a positive study that Sarah Hurwitz presented with, as you can see, improved objective responses and also about a 2-month improvement in progression-free survival with benefit in patients with brain metastases. Now, I think we have to wait for the overall survival results in this combination. I think the first-line setting, we all continue using the Cleopatra regimen. In most patients, I will use in second-line setting trastuzumab deruxtecan, but probably this can be an option for patients with brain metastases. Now, if we're gonna use tucatinib, I think we just don't know what combination is gonna be better, tucatinib with capecitabine and trastuzumab, or tucatinib with T-DM1 as presented in this trial. And I think we still have that big question regarding combination versus sequential single agents. Do we rather give 2 great drugs at the same time or use them sequentially? So I think we're still not ready to change practice, but we may incorporate some of this data. Now, in some of my last slides, I just want to acknowledge that advances also come not only for bringing new drugs, but also on improving the treatment delivery of the drugs that we already have. We need to find better ways to administer the drugs that we have access to and decrease the side effects of our drugs. This study was presented at FASCO, and in a very simple way, it evaluated if there's a better way to administer capecitabine. So it compared the standard dose by body mass index, 2 weeks on, 1 week off, for a fixed dose of 1,500 milligrams twice a day, 1 week on, 1 week off. That in practice is something that we all do when patients have toxicity. So in this small trial, patients with metastatic breast cancer were evaluated with these 2 schemas of capecitabine. PFS and overall survival were the same, but look please at the dramatic decrease in side effect. We improve serious side effects, we improve treatment adherence without affecting outcomes, but really maintaining the quality of life of our patients. So in summary, 2023 was a great year, and we continue to make substantial progress. There are today more drugs available to our patients, and while many questions regarding resistance and biomarkers remain unanswered, I think one of the big questions is sequencing and what is the optimal sequence, and I think we do not know this answer for all the patients. We need to design better clinical trials that answer more than one question and that really try to address sequence. We need to be open to these new algorithms that are going to be challenged by the incorporation in the early stage setting. We need to remember that we're treating with different tumors. Tumors have unique characteristics, but beyond that, we're treating with different patients. And we need to partner with our patients. I think this meeting has been key, highlighting the voice of our patients because they have the choice to understand the uncertainties of the data and to help us choose what side effects are relevant for them. So we need to engage in this shared decision. So as I close, I just want to finish acknowledging that I practice in a very privileged setting. I have access to all these drugs. And I know that that's not the same for all of you. So I think as we think about progress, we all need to continue working so all the patients everywhere have access to these advances. I want to thank to your attention and of course to BCRF and Susan G. Komen for funding my work, and Ilana Schlam, together with me, it's going to co-author an invited article at the Breast that it's going to summarize this presentation. Thank you so much.
Thank you so much, Mariana. This concludes our session. If you're interested in food and beverage, please turn to your left as you leave the auditorium, and in Stars and Night Ballroom 3 and 4, in 15 minutes we will have View from the Trenches. Thank you.